I've always been a sucker for challenges, and that's why Sir Rasco is my boss. And it's the same reason why I married a writer, is around. But never have I ever prepared myself for this challenge. This little man, Ellie, we call him Ellie, short for Elijah, is our ninja. And most times he's kind, curious, helps with the dishes, but there's that part of him that just makes me wonder if he's ousted the Lord of the Underworlds. He does that, yes. Now imagine if we are in the same room at the same time, I am checking your papers, and he wreaks havoc, and there's 3 of them. That's just a nightmare. So for now, no Ellie clones, and let's just talk about how we made him into this little creature. Let's have a brief recall of how nature creates you, living things. Okay? I did not create him through asexual reproduction. Oh, no, no, no. If you recall, cells are the basic unit of life, and in each cell, there's genetic material. That one. Now, the genetic material could be sequenced because they're composed of chemical units. Let's just give a letter for each one of them. For every species of living organisms, there's a specific sequence of these letters inside them. And if you're bacteria, and it's pretty simple, you could just split yourself and you create your own replicate. And it would have been wonderful if to see myself as my child, but no. There's another option. If you're a fungi or a yeast to the beer drinkers, you could also do budding. So I'd have to grow another cell on me, and then let it split off and then grow. But it wasn't that easy. There's a third option, and that's virgin birth. Now what happens in virgin birth? A cell, or like an egg, could develop into an organism. It would contain same genetic material as the parent, no other people involved, no other organisms involved. So if you're independent, try this one. However, I am a mammal, I am an animal, higher order being, highly complex, literally and figuratively. And so I have no choice but to do sexual reproduction. So sad. What's interesting with higher organisms is that we contain 2 sets of genetic information. And these 2 sets of genetic information needs cooperation, like what my boss said a while ago. So first, you need the egg cell, which contains a set of the sequences, and the sperm cell, which contains the other set. You have to merge them together, sometimes in a dim room, so that you create a wonderful individual that would resemble the same species that created them. So that's the catch. There's mixing of traits. In the previous type, it's just the parent, a bit narcissistic, a bit me, myself, and I. Now this is the whole point. What if sexually reproducing individuals like us really want to clone ourselves because we just love to? Or like I need a lot of people to check my exams or the thesis. Is there a way for me to actually replicate myself 1,000,000 times without having to find a mate? Any idea? Well, there is, and it's in biotechnology, and we call it cloning. So in cloning, the progeny or the children gets to have the same genetic material as the parent. I think I could replicate these on fleek eyebrows and have so many kids with these eyebrows. But no, this is artificial. The point is, in cloning, I can select the ones that I like and let them grow. And this is fascinating even for popular media. So if you recall Star Wars Episode 2, Attack of the Clones, a mercenary with the skills of a good warrior, a good fighter, was actually used. And he was the template, and there was an army of stormtroopers invading the galaxy. But there's one catch. These troops will obey without question, and a mother would love that. But the emperor was evil, and he used them to actually wreak havoc in the entire galaxy. So that's a bit scary. So while human cloning is basically in popular media, to date, there are no humans cloned. I would have loved one. But the idea is there are stem cells harvested from humans, and they use it for therapy. There are cancer clones from humans, cancer cell clones, that is, and they use it for drug discovery. And there was one successful attempt. But unfortunately, they found out that the cloned embryos that they used were actually fabrications, and it was not real. So you wouldn't be seeing a human clone in the next maybe 5 years, but that's just a very rough estimate. Also, UNESCO declared that there should be control on human cloning, specifically in their Universal Declaration on the Human Genome and Human Rights, Article 11, which says that those practices contrary to human dignity, such as reproductive cloning, shall not be permitted. It is unanimously agreed with 77 nations, including the Philippines. So no clones in the Philippines for the next years. It's a different story when it comes to animals. So there are 24 kinds of animals that have already been cloned, and they range from the wild mouflon, a gaur, and some of them are domesticated, like your dog, your cat, your goats, and your sheep. Oh, sorry. And a very famous sheep is? Very good. You have Dolly, who was named after the country singer, Dolly Parton. But Dolly, the sheep, not the singer, actually was euthanized 6.5 years after it was created because of lung complications and severe arthritis. There's also the concept of de-extinction. Now, scientists did not stop from Dolly. They tried other experiments, and they want to bring back those species that are extinct. The idea is you get a sample from their preserved tissues, and you bring them back to this modern world, like your dodo, Tasmanian tiger, or your favorite in Ice Age, Manny, the woolly mammoth. And it's nice if these are fluffy kinds. But what if it's the scary one? What if it's the dinosaur? You might not have known, but Jurassic Park is the predecessor of Jurassic World. And Jurassic Park at that time was scary. They got DNA sample from a mosquito. A mosquito accidentally bit a dinosaur, and this mosquito was trapped in a prehistoric amber, and they got out the blood, and there was dinosaur. It's not that easy, and the science is flawed. But with our advancement, who knows? A dinosaur might be knocking on your door. Well, there are still issues, because in animals, it's more of failures than success. The gaur, for example, died a few hours after it was created, so it's quite sad. And because they're animals, some of them are your pets, there are ethical issues. So nobody would want moral dilemmas when it comes to cloning. So no cloned animals mainstream perhaps next year, but they're considering twinning in pigs or cloning cattle to provide the demand on meat production of the world. But this is still like in the next years to come. For plants, it's quite a different story. They don't share the limelight. There's a lot of plants that have already been cloned, from your humble ornamental flower to your food crop to your trees. And yet, do they get the news? No. Did you ever see a tomato in the front page of your newspaper because it was cloned? No. And that was the point. There's not much emphasis in them. Probably because we eat them. We benefit from them. They're just pretty common. It's sad. Okay. So now I'm going to talk about micropropagation. Micropropagation is a general term wherein you could take parts of a plant, grow it in a controlled environment, and feed the world, use it for species propagation, and so on. Now, plant cloning falls under micropropagation. Specifically, one term, tissue culture. I'm sure you've heard about this. When you go to UP, you see tissue cultured plantlets. Now, in tissue culture, you have a plant. You look at for the parts that have dividing cells, and you cut it out. You chop them, put them in a jelly, much like what you eat. It has hormones, it has nutrients, and then you grow them inside an enclosed container, like a bottle, so that they will be disease-free, pest-free, and they'll have uniform growth. And it's wonderful. It's so easy. I could do it in the backyard, but not, because it's costly. It's expensive. You need this kind of equipment, and it makes sure that no disease comes in. You need to have a skilled technician, and she or he must have a green thumb. Trust me, those without green thumb, they don't have contamination, but they don't have growth. And you also need to make sure that everybody inside the controlled environment is happy. Because if the technician is heartbroken and she or he accidentally puts more sugar, there are changes, and that would not yield uniform growth. So, okay, there are natural clones, ma'am. Come on, it was mentioned. Plants can also do asexual propagation. Well, yes, like your potato. If you look at it closely, the swollen tubers, the ones that you eat for fries, hash browns, or potato chips, they actually have these small outgrowths, and the eyes could become new potato plants. So if one plant, for example, has 20 tubers, a family could save one tuber and plant it for the next season. That would feed the family. But what if your neighbors got jealous? Only you had the potato. I want more potato. So you need to increase the yield. And that's when nursery propagation comes in. You could cut out the stems, and then you'd have 300 clones for one plant. And that's in 2 months. You could feed the village. But you see, sometimes there are diseases, like the potato blight, and it wipes out a lot of your food source. What if you need to provide food for a number of people in a very short time possible? That's when tissue culture comes in, because one cutting could yield 100,000 clones in 6 months. Now imagine if you plant a cutting every day. Wouldn't that be enough to feed you? My God. But it's not really a perfect technology. If you remember, those plants inside the bottles, they are clones. But what if you isolate one and you try to get materials from it? You grow it, and unfortunately, the sugar brand was different. And you see there are changes. There's a pink color, or there's a stripe in the stem. Or what if you have another clone from the same batch, you grow it out, you plant it, but the technician got busted. And so he was so sad. And apparently, that sadness created variants again. And you now have different leaf patterns. So to the many, they would call this mutants, because they're not what they are as expected. But what's interesting is, some of them actually still contain the same genetic material, same sequence of the letters. It's just that the environment influenced you. So for this discussion, I'd like to propose we call them divergents. And what's good about divergents? They bring freshness. They bring novelty into the gene pool. Divergents are very, very good if you want to make elite lines of crops. For example, in coffee, of the 800,000 clones that they produced, they had 8,000 with divergence, or somaclonal variants, as we call it. And because of this, they were able to look at the possibilities of extending traits to create elite Arabica coffee. In a different study, blue wings, because these flowers used to just bear blue flowers with a white center, making it a wishbone. But through some somaclonal variation, or divergence, they were able to have white, purple, and pink flowers. And that's good, because you could sell them at a higher price. They're new. And I'm sure you're wondering, are there more common examples, products of plant cloning, perhaps those that I know? How about this one? Any guess? If you actually work hard in the mountains and you go home and feel that there's a thickening on your skin, you'd call it a callus. That's right. These are thickened plant clone cells called callus, calli in plural, and they're from carrots. My students are the ones who actually made that in the lab. Now, carrots are one of the long-maintained plant tissue cultured plants in the lab as early as 1939. And the success stories leads to many different kinds of carrots nowadays. And another one, any guess? These are bananas. Yes. So bananas are tissue cultured to meet the demand. It's now the second most loved fruit in the world, specifically the Cavendish cultivar. And it's next to citrus. So to meet with the demand, so that in winter or in summer, in El Nino or La Nina, you get your bananas, they tissue culture it. And what's interesting is the banana industry is so big, so the tendency is there's now more diseases because they have monoculture. But because of divergence, they were able to get some lines that are resistant to one particular disease, for example, Fusarium wilt. And because of these divergence, they were able to have resistance traits that were now you can mix with your clones. So it's interesting. And finally, I'm not sure if the younger generation knows this. This is Davao's Pride. This is the Waling-Waling, Banda Sanderiana, or Yuante Banderiana. And this is now extinct in the wild. You cannot see this in the forests of the Philippines, perhaps unless you go to for the jungles that have not been gone or that have not been explored. But what's good is tissue culture was able to obtain clones of these orchids. And so we can still keep the species. This sells at about 10,000 pesos in its fully grown flowering state before, but now tissue culture ones are cheaper. They're selling at a batch, about 100 a piece or less. So it's now for the masa. For UP Mindanao, because my boss is really persevering, we also want to do saving another species, and that is your Philippine pitcher plant. Now, the Philippine pitcher plant is endangered because it is over-collected from the wild. What's very interesting in this plant is this pitcher. It's a modified leaf. And these plants usually grow on tree trunks where there's less nutrients. For the plants to adapt, for them to have nutrients, what they do is they created a pitcher at the tip of their leaf, made it appear very bright and beautiful, so that insects and small animals could be attracted. And when they go near and they slip, they get to be digested inside the water. So it's termed as carnivorous plant. And together with Dr. Rasco and our cooperator, Ate Bilma, who happens to be here, Ate Bilma Fertes, please. Okay. We have the project on saving the Philippine pitcher plant because while it produces 1.5 liters of pitcher, it has the tiniest seeds you could imagine. It's like hair lice. And these seeds, when you just let them grow in the wild, takes a long time for them to reach that maturity stage. If there's overcollection, how can these species catch up with their demand? And so there was a brilliant idea. Let's clone them. So we took out leaf parts and we cloned them in the lab. These are not the actual pictures, but they resemble as this. So these are your calli from leaf, and given time, you could grow them in bottles, so you would have pitchers. And given more time, you could expose them outside, plant them in a media, and in this stage, you could sell them, say, half of it, half of your propagules. You could sell it in fairs, you get rich, and half of it you could introduce to the wild if you are kind. And that's what we did, because we're kind. So we gave half of it to Ate Bilma, and she took care of it, and the pitcher plants are now there in Marilog. Visit her farm. Okay, next is the nipa palm. Yes, these are the abandoned, unwanted, unnoticed plants that you might not even really know, but their leaves are harvested for roofing material. And they thrive in areas where there is low nutrients, but they house shrimps, crabs, and shells. So people who go to Digos area, perhaps, or near Talomo, Turil area, you would see these nipa palms. What makes it more interesting is this. The flower stalk produces a sugar sap, and you could harvest it for vinegar, or if you are, I don't know, you're lost, don't know where to go to, you could ferment it further, make it laksoy or vodka. So, mas maganda ang tama. You might probably wonder, why cloning, my dear? Well, because cloning is challenging. It raises issues. The process itself is not perfect. It opens possibilities. So cloning is challenge. And like I said, I like challenges. Bring it on. But cloning also makes me think of the responsibility. Makes me think of one man, but it's not my husband, I'm sorry. And it's not the Prime Minister who went to Jollibee. It's Kenshin Himura. He's Samurai X. Now in the story, Kenshin Himura is an assassin. But he had a change of heart. So he had the sword reversed so that the blade faces him. And it reminds him of his promise to be good and not kill anymore. And I like to see myself as the female version of Kenshin, the sexier one. The idea is, if I would treat cloning as that blade, as that sword, and be like an assassin, I could kill a life. But if I take responsibility and let it face me and take the responsibility of using it for the good, I could use it for therapy, for drug discovery, for saving, conserving species, for finding alternative fuels, and for helping feed and shelter the world. And it opens a lot of possibilities. Cloning is a challenge, and it's not just a challenge, it's a very big challenge. Thank you.